So this quick lecture is going to be just about test crosses, which I will go ahead and say up front. People make this very confusing and hard for themselves when really and truly it's not that difficult, okay? So look here, a test cross by definition is crossing an individual where you don't, as the person working the problem, you don't know the genotype, all right? So what you do know is that that individual has at least one dominant allele, so you know the phenotype. And so you're going to cross that unknown, quote unquote, individual with a known individual that's homozygous recessive. And so this is performed in an effort to determine the actual genetic makeup of an unknown individual. This is something, as you can see here, like a plant or an animal breeder might breed an unknown parent plant with a known one in order to figure out what the possible offspring are going to be. So for example, let's just say that I was a plant breeder, right? And that I'm breeding these plants, okay, that produce either purple or white flowers. And let's say that someone in class says, hey, Miss Zellmer, I brought you the type of plant that you breed, and it's a purple plant, okay? And that's all I know about that plant, right? So then I want to say, or I'm going to say to myself, I wonder what type of offspring this plant is going to produce. Because let's say that as that plant breeder, that I know that nobody really wants to buy white flowers, okay? Like nobody really likes them. Let's just kind of couch it that way, okay? And so what I don't want to do is spend a lot of time and effort breeding this plant if it's going to potentially produce white offspring, right? Or let's kind of flip that and say, let's say that white offspring, man, they are plants that if you can produce them, that people are going to pay a lot of extra money for them. And so maybe you're more likely to want to breed that plant to make more plants if you know you're going to make more money, okay? So this is pretty simple. Every single time you do a test cross, you're going to do 2 Punnett squares. And P is not a great letter, okay? But we've got our Punnett square over here where the parent plant that's purple is homozygous dominant crossed with this known homozygous recessive white plant. Then over here we've got our parent plant that's purple that's heterozygous, okay, with our known homozygous recessive white plant. Now in the grand scheme of things, this plant right here and this plant, those are one and the same because we just don't know what that second allele is in this plant because all we can see that it's purple, okay? And so then you're going to perform these 2 Punnett squares on paper, right? This is how we're doing this in this class. You know, the plant breeder's actually going to cross these 2 plants, move pollen from one to the other to produce new seeds, right? And so we've got, in this case, offspring have a 100% chance of being heterozygous with a capital and a lowercase p and making purple flowers. Then here we've got a 50-50 shot, a 1-to-1 genotypic ratio and a 1-to-1 phenotypic ratio. Okay, so if I were to write that here, I would write that my genotypic ratio is going to be this, okay, or for purple, right? So 100% of your offspring are going to be purple there. Here I'm going to have 2 to 2, which can reduce to 1-to-1. And then 2 purple to 2 white, which will reduce to 1-to-1. Okay, and your 2 to 2 and your 1-to-1, that's one and the same. Okay, both of those are correct answers, right? And then you're going to have to be able to explain to me why you would do this. Okay, and the reason that you're doing a test cross is that as in this case, as the student, you don't know what that second allele is. All right. If you have any questions about test crosses, let me know.